Today is August 28th, 2023. The time is 10:27 a.m. I'm going to read an article from the National Library of Medicine, and it's dated October 3rd, 2022. The title is Coronavirus COVID-19 Vaccine-Induced Immune Thrombocytopenia. Coronavirus disease 2019, COVID-19. The illness caused by severe acute respiratory syndrome, coronavirus 2, continues to have significant morbidity and mortality worldwide, with many nations enduring multiple outbreaks of this viral illness. Many vaccines, BNT162b2, messenger RNA, 1273 AD26.COV2.S and CHADOX1 in COVID-19 have been developed at an unprecedented speed using distinctive technologies to prevent COVID-19. Several cases of uncommon thrombotic events associated in combination with thrombocytopenia were observed in patients who were vaccinated with CHADOX1 nCoV vaccine, COVID-19 vaccine. This article will highlight the role of the interprofessional team in the management of patients with this rare syndrome of vaccine-induced immune thrombotic thrombocytopenia, VITT. Objectives: review the epidemiology of VITT. Describe the pathophysiology and pertinent clinical features of VITT. Review the treatment options in management of VITT. Describe the importance of interprofessional approach in managing patients affected with VITT that would lead to better patient care and improved outcomes. Coronavirus disease 2019, COVID-19, the illness caused by severe acute respiratory syndrome, coronavirus 2, continues to cause significant morbidity and mortality worldwide, with many nations enduring multiple outbreaks of this viral illness. Besides the importance of infection control measures to prevent or decrease the transmission of SARS-CoV-2, the most crucial step to contain this global pandemic is to vaccinate individuals to prevent SARS-CoV-2 infection in communities across the world. Many vaccines have been developed at an unprecedented speed using distinctive technologies to prevent COVID-19. Vaccination triggers the immune system, resulting in the production of neutralizing antibodies against SARS-CoV-2. Four vaccines, namely the BNT162b2, messenger RNA 1273 AD26COV2, and CHADOX1 nCoV-19 have been approved to prevent COVID-19 in many nations worldwide, including the United States. The BNT162b2 messenger B this is one vaccine. The BNT162b, messenger RNA 1273 have been approved by the US FDA, and the AD26COV2 has received emergency use authorization. One exception is the CHADOX nCoV vaccine, which has not yet received emergency use authorization or approval from the U.S. Food and Drug Administration, FDA, for the use in the U.S. The BNT162b2 and messenger RNA 1273 vaccines are both messenger RNA based, while the AD260 or COV2S and CHAD vaccines incorporate replicon competent adenoviral vectors in them. Let me make this a little bit clearer. So, what we're talking about is the different technologies, the distinctive technologies. We're talking about distinctive technologies to prevent COVID-19, and these are the vaccines. We have uh, we have the BNT vaccine technology, and what this technology is, this is messenger RNA technology. That's the technology that is used. Moderna's vaccine messenger RNA 1273, that the technology is messenger RNA, synthetic messenger RNA. The AD26CoV2S, that's the Johnson and Johnson vaccine. And the technology used there is viral vector. And in this case, it's the adenoviral vector technology. We're going to talk about that. And then here we have the AstraZeneca vaccine. Again, this is a adenoviral vector vaccines. And these two vaccines here are adenoviral vector vaccines. And a safety study was Jesse Gelsinger, a participant in a safety study on this technology, viral vector technology, died in a safety study. This is known, Jesse Gelsinger's death is known to the FDA. Why the FDA would approve an adenoviral vector technology after the death of Jesse Gelsinger. And what we're talking about here is coronavirus vaccine-induced immune thrombotic thrombocytopenia. And I'm going to just keep reading. In late February 2021, a new clinical syndrome, let's highlight that, let you know that this is a new clinical syndrome characterized by thrombosis at atypical sites combined with thrombocytopenia was observed in multiple patients days after vaccination with the ChAd vaccine. This is the viral vector, adenoviral vector. In April 21, similar clinical sequelae were reported in patients after vaccination with the adeno26-CoV2 vaccine. This is Johnson & Johnson's adenoviral vector technology vaccine. We're talking about vaccine technology. What vaccine technology? Adenoviral vector. Preceding the approval of these vaccines, the clinical constellation of this new syndrome, we're here talking about a new syndrome, was not observed in clinical trials of I'm going to say ChAd because that's what it kind of looks like to me, the word. The ChAd vaccine, again, we're talking about adenoviral vector technology. And a single case was observed in the AD26CoV, which is the Johnson & Johnson adenoviral vector vaccine technology. Further, it says vaccine and a single case was observed in the Johnson & Johnson vaccine trial recipient. Furthermore, the incidence of major adverse effects has remained exceptionally low following the vaccination. Now, furthermore, the incidence of major adverse effects has remained exceptionally low following the vaccination of more than 400 million people worldwide. This novel clinical syndrome, we're talking about a new syndrome, demonstrated striking similarities to heparin-induced thrombocytopenia. However, in the absence of prior heparin exposure, was named vaccine, let's make that clear, vaccine-induced thrombocytopenia. It is also known as vaccine-induced prothrombotic immune thrombocytopenia in European nations and Canada. Conversely, in millions of doses of this is again the Pfizer BNT vaccine and the Moderna vaccines administered so far, this clinical syndrome has not been reported. Due to the concern of VITT associated with, now this is the Johnson & Johnson vaccine, the FDA modified the emergency use authorization and recommends limiting the use of this vaccine to only individuals greater than 18 years of age who are otherwise ineligible to receive any other FDA-approved vaccines due to anaphylaxis to messenger RNA vaccines or its components, or are unable and unwilling to receive any other vaccine. Per the American Society of Hematology, vaccine-induced immune thrombocytopenia is defined as a clinical syndrome by all of the above described abnormal of the below described abnormal laboratory and radiological abnormalities occurring in individuals 4 to 42 days after vaccination with the Johnson & Johnson or the AstraZeneca vaccines. Development of thrombosis at uncommon sites includes cerebral venous sinus thrombosis, splenic venous thrombosis, mild to severe thrombocythemia. However, a normal platelet count does not exclude the possibility of this syndrome in its early stages. Positive antibodies against platelet factor 4, PF4, identified by enzyme-linked immunosorbent assay, ELISA, significantly elevated D dimers greater than 4 times ULN. This review article aims to describe the etiology, epidemiology, pathophysiology, clinical features, diagnoses, and management of COVID-19 vaccine-induced thrombotic immune thrombocytopenia based on the latest available published literature. Several cases of uncommon thrombotic events associated in combination with thrombocytopenia were observed in patients who were vaccinated with the AstraZeneca vaccines. Although a causal relation has not been well defined, the increased reporting in the United Kingdom, as well as in Europe, of cardiovascular or CVST, I don't know what it means, and thrombocytopenia in individuals vaccinated with AstraZeneca vaccine compared to the absence of cases in individuals immunized with Pfizer or Moderna vaccines proved to be a strong indication that this syndrome was potentially associated with the AstraZeneca vaccine. Due to the rarity of this clinical syndrome among vaccinated individuals, there is currently no evidence suggesting that individuals with prior history of thrombosis or risk factors for developing thrombosis are at increased risk of developing vaccine-induced whatever, VITT. It is also unclear at this time as to why this immune-mediated thrombosis manifests mainly in the cerebral vessels and splenic circulation. Epidemiology. The incidence of VITT extremely low. The true causal relationship between this new clinical entity and the vaccines has not been established yet, but is considered plausible based on a statement issued by the World Health Organization on April 27, 2021. As per the European Medicine Agency, the incidence of VITT after vaccination with AstraZeneca vaccine is estimated to be 1 in 125,000 and 1 in a million. Notably, more than 80% of the patients in reported cases of VITT were females and aged between 20 to 55 years of age. After a brief pause in the administration of AstraZeneca vaccine, the UK Medicine Healthcare Products Regulatory Agency and the European Medicines Agency, EMA, completed internal reviews and favored resuming vaccination with this vaccine, emphasizing that the benefits of the vaccine outweigh the risk of developing thrombosis. However, the causal relationship was not described by either agency. Recently, of the nearly 6.8 million doses of AstraZeneca's Johnson & Johnson's vaccine, a small number of female patients aged between 18 and 48 developed similar clinical constellation of cerebral venous sinus thrombosis and thrombocytopenia that occurs 6 to 13 days after vaccination. The CDC and the FDA have recommended a pause in vaccination with Johnson & Johnson vaccine in the US to allow for further review of this vaccine. The pathogenesis of vaccine-induced thrombotic thrombocytopenia is not known at this time. However, given its clinical presentation and biochemical similarities to heparin-induced thrombocytopenia, which is a prothrombotic and potentially life-threatening condition, a similar immune-mediated response induced by these adenoviral vector vaccines has been postulated as immune complexes with a mixture of antibody specificity similar to HIT was noted in the serum of patients who developed this syndrome. Based on a report by the Society for Thrombosis and research, it has been hypothesized that post-vaccination antibodies are formed against platelet antigens as part of the immune stimulation process. These antibodies trigger massive platelet activation against PF4, resulting in immune thrombotic thrombocytopenia. Cross-reaction between the vaccine and platelets of PF4 has been highlighted as a potential contribution factor in the pathogenesis of this syndrome, especially knowing that adenoviruses can bind to platelets, resulting in platelet activation. Further clinical studies are needed to determine the pathophysiological mechanisms of this new syndrome precisely. A detailed clinical history regarding the onset and duration of symptoms, vaccination history that includes exact timing, drug history, and particular recent exposure to heparin should be obtained by treating providers. Pertinent clinical features concerning VITT: new neurological symptoms such as severe, recurrent, or persistent headaches, seizures, blurry or double vision, focal weaknesses, aphasia, severe abdominal pain or persistent nausea and vomiting, backaches, shortness of breath, chest pain, leg pains, and or swelling. I don't know how to pronounce that. Evaluation. Laboratory assessment in VITT. Based on the treatment guidelines by the British Society of Hematology, BSH, Society of Thrombosis and Hematology Research, GTH, and American Society of Hematology, ASH, patients suspected of VITT should undergo initial laboratory workup and imaging modalities as described below. CBC with platelet count and peripheral blood smear. Typical platelet data range from 9,000 to have been reported. I won't go into read all of this. The American Society of Hematology treatment recommendation for VIT is similar to that of severe HIT. In patients presenting with thrombocytopenia, documented evidence or suspicion of thrombocytopenia positive or pending PF4. Considering this newly described syndrome is evolving, the prognosis of vaccine-induced thrombocytopenia is not known. The prognosis of vaccine-induced thrombocytopenia is not known. This estimated mortality rate related to the use of AstraZeneca vaccine was calculated to be low at one death in a million. However, considering thrombosis more commonly involves atypical sites such as cerebral venous rapid adhesion is critical.